Okay, here we are, chapter 9, and we are going to talk about cellular respiration. So, this is a pretty detail oriented lecture and topic, so make sure you take lots of great notes, draw pictures, stop, pause the video so that you can draw what you need to. Okay, here we go. Okay, so we're going to talk about anaerobic and aerobic pathways. So anaerobic means no oxygen, and aerobic means with oxygen. I always think of if you're doing aerobics, then you have to breathe a lot because it's hard. So go ahead and draw this picture, pause the video and draw this picture. And if you want, you can take notes on the picture or you can just take them under the picture. So go ahead and pause it. So when oxygen is not present, this is your anaerobic pathway, and it goes into fermentation, and we'll talk about this at the end of the lecture, but you make lactic acid or ethanol. We're going to spend most of our time talking about when oxygen is present because that's what our cells do the majority of time. And then you end up getting this huge amount of ATP. Okay? So, the main types are, like I said, anaerobic and aerobic, and just kind of some ideas of them is anaerobic evolved first because if you remember, the Earth did not have oxygen in the atmosphere until after photosynthesis. So anaerobic happened first, doesn't require oxygen. It starts with glycolysis, just like in aerobic, and it's completed in the cytoplasm or cytosol, as you might see. So aerobic pathways evolved after because we need oxygen. So after photosynthesis happened, it also starts with glycolysis, but the end happens in the mitochondria. So this is a great picture that I would also like you to draw because one of the things you're going to need to be able to do is what I call the ins and outs. So what are the reactants that go into the different processes and what are the products or the outs that come out because everything is linked. So in glycolysis, you can see that you have some ATP going in, but you have also ATP coming out. You can see the product of pyruvate, which ends up going into the Krebs cycle. You have all of these electron carriers we'll talk about in a little while, and they deliver their electrons to the electron transfer transport chain or ETC, and then you have this huge amount of ATP. So it's not important that you know the numbers of all of these different chemicals. You need to know that they're produced and their function. You also need to know that this electron transport chain is the moneymaker. It's what makes all the ATP, or almost all of the ATP, that we get out of a single glucose. So, some words that you need to know, and you probably, hopefully, remember these from chemistry. So, if you say a molecule was reduced or oxidized, I think one of the chemistry teachers taught me to remember oil rig. So, oxidation is losing an electron, reduction is gaining an electron. So as glucose gets oxidized, so through cellular respiration, glucose loses electrons, and those get put onto the coenzymes, the NAD and FAD that you saw in the slide before, and they take those electrons to the electron transport chain. So that's kind of the gist of the whole process. So we'll start with each of the different parts of cellular respiration. So our first one is going to be glycolysis. And it's important that you know kind of the purpose and also where it happens. And that, like I said, what goes into the process and what comes out. So there's two different steps. The first step is endergonic, so it requires energy. And so if you look down here, you have two ATP that are going into glycolysis, and that activates glucose. And glucose, remember, is a 6-carbon sugar. And then what happens is the energy releasing step, so it's exergonic, and you actually get 4 ATP produced, so you have a net of 2 ATP. And also what's produced is electrons are getting pulled off of glucose and they get put onto NAD plus to reduce it and form NADH, which I call NAD. And then you also make two pyruvates. And pyruvates are a 3-carbon sugar because, so glucose is a 6-carbon sugar, and then you split it in half and you get a 3-carbon sugar. This is considered an anaerobic process because it does not require oxygen, so all living things do glycolysis. Now, some places you might see cytoplasm referred to as cytosol, so just so you know, it's the same thing. Okay, so the second stage, we move into the mitochondria. So there's 2 parts here. There's what's called a preparatory reaction, and that's just basically taking pyruvate and turning it into acetyl-CoA is what that's called. And you also produce a molecule of carbon dioxide here, so you end up getting a 2-carbon sugar, and then you also get an NADH as well. And then the Krebs cycle is the next part, and what happens is acetyl-CoA is oxidized again to form carbon dioxide. So if you think of cellular respiration, and one of the main products is carbon dioxide, so what you breathe out, this is where it happens. You also get NAD and FAD are reduced, so they pick up electrons in this process, and they take their electrons to the electron transport chain. And then a few ATP are also formed. So here's a picture of the preparatory reaction and the Krebs cycle. So you can just see that if you think of the overall reactants, you get pyruvate, and these little balls are the numbers of carbon. So you get 3-carbon pyruvate, and then out you get carbon dioxide. So here's your carbon dioxide right here. And so then the coenzyme comes in, and you get acetyl-CoA. And then you also get a NAD that's produced. And then the acetyl-CoA comes over here to the Krebs cycle, and it's a cycle because it regenerates the chemicals. And then here, important is that you get all of these electron carriers get their electrons because they're going to take them to the electron transport chain where all of ATP is used. And then you also get a couple of ATP here. So the coenzymes, NAD and FAD, accept electrons and hydrogen, so they're reduced. So coenzymes are not actually proteins, they are nucleic acids. It's kind of confusing. Co means that they are helpers, and so they're not enzymes, and that's why they're not proteins, they're nucleic acids. So they grab electrons, they get reduced, and then, like I said, I keep saying this because it's so important, their purpose is to take those electrons to the electron transport chain where you get lots and lots of ATP. So here is the electron transport chain, and we'll kind of go through this picture, and then I'll have the next slides to give you some words. So here are your electron carriers, NAD and FAD, and so what they do is they give the electrons to the electron transport chain, and as they go through these proteins of the electron transport chain, their energy allows hydrogen ions to be pumped into this intermembrane space, and so you form what's called a proton gradient. And the reason why that's important is because then those hydrogen ions diffuse down this protein called ATP synthase, and this is where you make all your ATP. The other thing that's really important here is that, so here's your electron getting taken off of NAD and then of NAD and FAD, and it goes through here. And then what happens to this electron is that it bonds with oxygen and some hydrogens to make water. So oxygen is the final electron acceptor, and that's super important. This oxygen right here is why this is called aerobic respiration. So another term for the ATP being made is called oxidative phosphorylation, and that happens, like I said, in the electron transport chain. So more specifically, it happens in the mitochondria on the inner membrane. So remember, mitochondria have a double membrane. They have their outer membrane, and then they have an inner one that's all like convoluted. So the coenzymes NAD and FAD donate electrons to the electron transport chain. So if they're losing electrons, they're being oxidized. And that's why it's called oxidation right here, oxidative. And then those high energy electrons enable the active transport of those hydrogen ions across the membrane to form that concentration proton gradient. And this process is called chemiosmosis. So the hydrogen ion gradient that's formed by the electron transport chain is called chemiosmosis. That's another really important word. When you have that electrochemical gradient of all of those protons in the outer compartment, they can flow down their gradient through this protein called ATP synthase to make ATP. And that's the phosphorylation part of oxidative phosphorylation, because what's happening is you're adding a phosphorus to ADP to make ATP. So how come oxygen is so important? This process only happens in the presence of oxygen because of this statement right here. So oxygen withdraws the spent or low energy electrons from the electron transport chain and combines them with protons or hydrogen ions to make water. So without the oxygen, then those electrons would just get kind of stuck in that chain and everything would back up and it wouldn't work. So when you don't have oxygen, we'll learn that some other things happen. So again, here's another picture, just a slightly different cartoon. So here is your NADH donating its electron, and as it goes through these proteins of the electron transport chain, hydrogen ions are going to get pumped up to this inner membrane space, and the energy from the electrons is what allows for this pumping action. And then here is your electron that's getting grabbed by an oxygen and a hydrogen to make water. And then over here is your ATP synthase. So now the hydrogen ions or protons go down their concentration gradient, and as they go down, they cause ADP to combine with phosphorus to make ATP. So all organisms that have an electron transport chain, in eukaryotic cells, it's located on the inner membrane of the mitochondria. And on the chloroplast, we'll see the electron transport chain is located on the inner membrane of the thylakoids. Prokaryotic cells, we've talked about this before. If anything is located on the membrane of an organelle, it's located on the plasma membrane in prokaryotic cells. So some prokaryotic cells have electron transport chains, but they're slightly different, and they don't make nearly the amount of ATP. So what is the efficiency? Why is aerobic respiration so important? So from one glucose molecule, you get 36 ATP. However, most of the energy is lost in heat. So when the hydrogen ions don't make ATP, the gradient releases heat. And another vocab word that's important is endotherm. So you are an endotherm, which means that you regulate your body temperature from within. And so this release of heat from all of these hydrogen ions being concentrated on one side of the membrane allows us to regulate our body temperature. Okay, so let's talk about some anaerobic pathways. So these do not require oxygen, that's why they're anaerobic. They produce less ATP, a lot less. And there's two that we're going to talk about. We're going to talk about fermentation pathways, and those include lactic acid and alcoholic fermentation. And then there's also the anaerobic electron transport chain. Okay, so if you think about it, which part of aerobic respiration doesn't require oxygen? And that's glycolysis, which is why all living things do glycolysis. So here are the two examples of fermentation. So animals like you do lactic acid fermentation, and you might have, you probably all felt this before, where your muscles start to burn. And what's happening is you're building up lactic acid because your cells themselves aren't getting enough oxygen. So you're still breathing, but your cells aren't getting enough oxygen. And so what happens is pyruvate, instead of going into the Krebs cycle, turns into lactate and then lactic acid. And it's important, this extra step, because we need to regenerate NAD. If we don't regenerate NAD, then glycolysis can't happen and we can't get the little bits of ATP. So the two steps here on the lactic acid fermentation and alcoholic or ethanol fermentation, the second step is to produce NAD plus so that it can regenerate and go back up here and get reused so that we can eventually make a few ATPs. So this is a quick way, a quick process, but it just doesn't produce as much ATP. So they both have glycolysis. That's one thing they have in common. And the NAD regeneration happens so glycolysis can keep making ATP. So I just put a picture of some sprinters because when you're sprinting or you're using lots of energy really fast, this is when lactate fermentation happens. So if you're a sprinter or you're doing something like weights, you've definitely felt the burn, and that's what's causing it. And then you can see here that your muscle cells are different colors, and we'll talk about that. What happens is in your cells you have what's called myoglobin. Myoglobin is similar to hemoglobin, so it carries oxygen, but myo means muscle, so it carries oxygen to your muscles. So the cells that are really dark have lots of myoglobin and lots of oxygen. So those would be doing aerobic respiration. The cells that are really light have less myoglobin, and so those would do anaerobic respiration more often. So if you're a sprinter, you have more of the light colored cells because you do more of the fast energy for short periods of time. The light cells have less myoglobin, but they have more actin and myosin, which are the filaments that make your muscles strong and big. And then, since we're getting right close to Thanksgiving, I wanted you to think about AP biology while you're eating your turkey. And so think about these questions and we'll discuss why they are related to anaerobic and aerobic respiration. So think about to yourself, what type of meat do you prefer, dark or white? I personally like the dark meat better. So think about why it is dark or white. So it's dark or white because of the amount of myoglobin. So the dark meat has more myoglobin and the white meat has less. So what type of activity is more common in dark or white muscles? So if you think of the white muscles, like the main one that we eat of a turkey is the breast meat, right? And that's attached to that when turkeys, I don't know if you guys know, but they don't fly a whole lot, right? They have short bursts of flying. They actually roost in trees. I was in Michigan when I was doing my master's, and we were hiking, and it scared the crap out of me that I was walking on this trail in the morning, and this turkey flew out of a tree. I had no idea that they slept in trees. But anyway, so their activity in their breast meat is very short and quick and strong, and so that's why it's white. However, if you look at the leg meat, right, leg meat is dark, and turkeys walk most of the time, and so they need to be able to have a lot of energy from those muscles, which is why it's dark. Okay, so this question, why is dark meat more fatty, right? If you think of it, it's less dry, it's more fatty. Well, the fat is energy. So the Dark meat has a lot more energy close to it because it needs it more often, and so it has a good supply. And then if you think about, you might have heard the term slow twitch and fast twitch. Well, this is the same kind of thing. Slow twitch would be those muscles that are used a lot and have more aerobic respiration going on. And then fast twitch would be the ones that have the really strong contractions but can't last very long. So sprinters have more fast twitch fibers and marathon runners have more slow twitch. So when you're eating your turkey, you can think of AP biology. Okay, last slide. Just a quick thing on aerobic electron transport. So this is carried out by certain prokaryotes. So the electron transport is on the plasma membrane or the cell membrane, and the final electron acceptor is not oxygen like it is in our cells, but it's something else, some other compound from the environment like nitrate. And ATP yield is really low. It's not the 36 that you get from aerobic respiration, but it's just another path that some prokaryotes do. So there was a lot of vocabulary, a lot of detail. So what I would do is I would actually watch this again, have your notes out and add to them, and make sure you draw all the pictures and take notes on them. Okay, have a great day. We'll see you later.